In this video, we will discuss about the effect of high-fat diet on the interactions of the lung and fibronectins. So, what it is, they check the platelet expressions on the high-fat diet and the control diet, and they did their studies on the mice model. So they checked whether the platelets are activated in the lung area or not. And in order to check that, what it is, they use the p-selectin markers. And what they are able to find out in the high-fat diet mice, they see more activated platelets in the lungs compared to the control case. And these things are quantified in the panel B, where we can see that the p-selectins stainings is significantly higher in the high-fat diet compared to the control. Then they check the mice models for the presence of these platelets in the lungs, and they are able to see that in the high fat diet they are able to see this platelet formations compared to the control case, and these things are again quantified in the panel D, which we can see that the high platelets formations are there in the high fat diet compared to the control. Thereafter, they wanted to check whether the fibronectins are also present, because before going into this so once they confirmed that there is a high activated platelets are formed in the high fat diet in the lungs. Now they wanted to see the reasons, the difference in the signaling pathways that are occurred in the high fat diet and in the control diet. So they did the RNA-seq analysis, and from the RNA-seq analysis they are able to find out that six of the proteins are highly upregulated, and out of these six, they see that fibronectins is also upregulated, and previous studies have also pointed out that fibronectin plays a role towards platelet activations. So their focus is now on the fibronectin expressions in those two conditions. So when then they stay in the two different mice models, uh, one with nude mice and one with high fat diet mice, and they're able to see that high fat diet mice has more fibronectin formations compared to the control, and these things are quantified in the panel H. That is, the fibronectins formations stainings is significantly higher in the high fat diet compared to the control. Now they wanted to check this thing using Western blot, so they inject their PBS into the mice, and then in the inject PBS is conditions is been called here as perfused lungs conditions, and a non-perfused lungs condition in which no PBS is introduced. When no PBS is introduced, they are able to see more presence of fibronectins formations, but when PBS is introduced, they are able to see less fibronectins formations, indicating that the fibronectins has a more circulation rate. Fibronectin has a more circulation rate, as when we perfused it, it will with the PBS, the fibronectins might goes into the circulations. That's the reason we see less PBS formations in the perfused conditions compared to the non-perfused conditions. And these things they have quantified in the panel J, where we can see that FN1 expressions relative to HSP90s are significantly higher in the high fat diet compared to the control, but in the case of perfused conditions we do not see a significant difference. The difference is non-significant. So this indicates that there is, because of the circulation rate, the fibronectins can be circulating and in the non-perfused conditions we can see the high fibronectin formations, which is usually the normal conditions in the mice. And thereafter they check this fibronectin presence in the plasma and the platelets, and to find out what is the source of these fibronectins. In the plasma they are not able to see a significant difference. You can see that the P levels is greater than 0.05, is 0.053, but in the platelets they are able to see a significant difference in the HFD conditions compared to the control conditions. These things quantified in the panel N, where we can see that the fibronectins are more present in the platelets compared to the control case, more present in the platelets with HFD conditions compared to the nude mice conditions. Now they wanted to check the activations of the platelets, whether they are more prone towards activations or not. So for that, in the HFD conditions, so in the HFD conditions, when they check it with and without tryptin, they are able to see in both the conditions the activations probability of the platelets in the HFD conditions is higher compared to the control case. And these things they have quantified in the panel P and Q. In the panel P, it shows about the FN FITC with tryptin blue conditions, where we can see that with tryptin blue conditions, indicates that the internalized part of the cells, and it shows that it is significantly higher in the HFD conditions compared to the nude mice conditions. Tryptin blue has a property that it can enter the cells whose membrane is damaged. And when they check for this, check the MFI, mean fluorescence intensity, and they can see that mean fluorescence intensities are also significantly higher in the HFD mice conditions compared to the control conditions. And so all this in together indicates that in the high fatty diet there is increased interactions between fibronectins and the platelets, and that might cause the pre-metastatic niche. And this is the panel C also not only represents the p-selectins, it also represents the pre-metastatic niche conditions or formations, pre-metastatic niche formations. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.